Hey everyone, and welcome to The Explainer. So, if you're a high school biology student and you're feeling that, familiar knot of test anxiety in your stomach right now, just take a deep breath. You are in the exact right place. Today, we're basically transforming Dr. Usama Welbohantallah's complete high school biology study guide into your absolute ultimate exam roadmap, where you're going to break down the hardest questions, navigate those really common student traps, and build some serious exam confidence. Okay, let's dive right into this. Lowering your cognitive load is literally step one to studying effectively. So here's exactly what our syllabus looks like today. We've got 1. Cell biology, 2. Cell division, 3. Genetics, 4. Molecular biology, and 5. Evolution. Let's do this.
Starting off with Chapter 1, Surviving Cell Biology. Our mastery checklist goals here are straightforward. Predicting water movement with absolute certainty and effortlessly distinguishing between active and passive transport.
Now let's do a quick pitfall pivot around osmosis. Look, students constantly mix up hypotonic and hypertonic environments, and it costs them massive points on exams. So, the golden rule you absolutely must remember is this: water always moves toward the higher solute concentration. Think of it like this. If you put a cell in a hypertonic, highly salted solution, water is going to rush out, and that cell is going to shrink.
To make sure you never freeze up on a test question, the guide gives us this super reliable 5-move problem-solving strategy. First, identify if the cell is prokaryotic or eukaryotic. Second, match the organelle to its function. Third, use tonicity to figure out water flow. Fourth, check if we're dealing with active or passive transport. And fifth, apply surface area to volume logic. This exact framework will help you solve pretty much any cell bio problem they throw at you.
So let's put that 5th step to the test with a hard-tiered practice question pulled straight from the guide. Calculate the surface area to volume ratio of a cube-shaped cell with a side length of 2 micrometers versus 4 micrometers. Now don't panic at the math here, you've totally got this.
Let's just break down those numbers. For the 2 micrometer cell, your surface area is 24, and the volume is 8, which gives us a ratio of 3.0. But for that 4 micrometer cell, the surface area jumps up to 96, and the volume hits 64, dropping that ratio down to 1.5. And this brilliantly illustrates the core concept. As size increases, the ratio falls. It proves that larger cells exchange materials way less efficiently.
And that rule of efficiency transitions us perfectly into Chapter 2, Mastering Cell Division. Your mastery goals here are correctly ordering the phases of mitosis, prophase, metaphase, anaphase, telophase, or PMA, and explaining the crucial difference between the products of mitosis and meiosis.
A super common mistake here is mixing up mitosis and meiosis I when you're looking at diagrams. You've got to treat those visual cues like insider information. Always look for the single line of chromosomes at the cell equator for mitosis versus pairs of homologous chromosomes lining up together at the equator for meiosis I.
All right, here is your challenge question to test your ability to track chromosome numbers through these complex division phases. A cell with 12 chromosomes undergoes meiosis. How many chromosomes are in each cell after meiosis I and after meiosis II? Think carefully about what exactly is separating during each stage.
Got your answer? It's 6 chromosomes after meiosis I and 6 chromosomes after meiosis II. Here is the exact logic from the answer key. Homologous chromosome pairs separate in meiosis I, which halves your total count from 12 down to 6. Then, in meiosis II, it's the sister chromatids that separate. So the overall chromosome count stays at 6, but they're now single chromatids instead of duplicated ones. Makes sense, right?
Let's zoom in from dividing cells to the actual code inside them with Chapter 3, Cracking Genetics and Heredity. Your checklist items to ace this section means setting up those massive 16-box dihybrid crosses and successfully reading inheritance patterns off of family pedigrees.
First off, it is absolutely crucial to distinguish between genotype, the actual hidden alleles, and phenotype, the visible trait you can see. But I really want to pivot to a major trap: sex-linked traits. Remember, guys, males express recessive X-linked alleles much more easily. Why? Because they only have one X chromosome. There's no dominant allele on a second X to mask it.
If you ever find yourself freezing, just staring blankly at a Punnett square on test day, use this step-by-step pattern recognition framework. Step 1, write out the parent genotypes and their gametes. Step 2, build the square. Step 3, carefully read your genotype, then your phenotype ratios. Step 4, check the inheritance pattern. And Step 5,
Remember, you can always use a test cross with a homozygous recessive to figure out an unknown genotype. Okay, let's actively apply that strategy to an X-linked recessive question. A colorblind man marries a carrier woman. What is the probability that a son will be colorblind? What about a daughter? Take a split second and think about how you'd set that up. If you followed the framework, the logic reveals that since the mother is a carrier, she passes the affected X to half her sons, making 50% of the sons colorblind. For the daughters, well, they automatically get the affected X from their colorblind father, and they have a 50% chance of getting the affected X from their mother. So daughters are 50% colorblind and 50% carriers. Boom. Easy when you break it down. Next up on the agenda is Chapter 4, Navigating Molecular Biology. To master this unit, you must be able to confidently apply base pairing rules across DNA and RNA, and correctly classify different types of genetic mutations.
Here is a classic student pitfall that happens literally all the time on finals. When you are asked to transcribe a DNA sequence into an mRNA strand, please do not accidentally write down thymine, or T. RNA uses uracil, or U, to pair with adenine. Just remember, in RNA, there is absolutely no T time. It's a tiny detail, but it costs students a lot of points.
Let's test your central dogma knowledge with this one. A point mutation changes the DNA base from C to T. The resulting mRNA codon changes from CGU, which codes for arginine, to UGU, which codes for cysteine. Classify the mutation and predict its possible effect.
So, because this mutation alters a single specific amino acid in the final protein sequence, it is classified as a missense mutation. As for the effect, well, it can range drastically. Depending on exactly where it happens, it could be completely harmless, or it could cause a severe condition, like sickle cell anemia, by dramatically changing how that protein actually functions. We are in the home stretch now, as we tackle Chapter 5, Conquering Evolution Exams. Your goals here focus heavily on applying the Hardy-Weinberg equation correctly, and distinguishing between directional, stabilizing, and disruptive natural selection.
The biggest Hardy-Weinberg trap is honestly just panic. Do not freak out at the formulas. Just remember the core rule, P plus Q equals 1. If you're given a percentage of a population, always solve for your basic individual allele frequencies, P and Q first, before you do anything else.
Here is your final challenge. If you can solve this hard question, you are absolutely ready for the exam. A population has allele frequencies P equals 0.7 and Q equals 0.3. Calculate the expected frequencies of all three genotypes under Hardy-Weinberg equilibrium.
Let's walk smoothly and slowly through the math to show how completely manageable it really is. For the homozygous dominant, you just square P. So 0.7 squared is 49%. For heterozygotes, the formula is 2 times P times Q. So 2 times 0.7 times 0.3, which gives us 42%. And finally, for homozygous recessive, just square Q. 0.3 squared is 9%. 49, 42, and 9, they all add up to 100%. Perfect!
So, the real question is, are you ready to ace your biology finals? Think about the massive advantage of walking into your test armed with these exact frameworks, knowing exactly where the traps are hidden before you even sit down. We have only scratched the surface today. I strongly urge students, parents, and teachers to download the full 131-page guide. It's got all 360-plus questions, mastery checklists, and incredible visual summaries. You can find the link to the Bow Team publication store right down in the description. Get the guide, trust your strategies, and go conquer that exam. Because once you understand the rules of life on a test, you start seeing them everywhere in the real world. Good luck out there.